Hello everyone and welcome to today's webinar, Get to Insights Faster and Easier with AI Image Analysis Tools. I am Cassie Soltman of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar. Dr. Yung Choi is the Product Owner and Manager in Data and Analysis at Leica Microsystems. With a Ph.D. from Columbia University in dopaminergic circuitry and years of experience in microscopy and image analysis, Dr. Choi brings deep expertise from both research and leadership roles to drive smarter, AI-powered tools for accelerating scientific discovery. Dr. Choi, you may now begin your presentation.
Hello, my name is Yung Choi and I'm the Product Owner and Manager for Data and Analysis at Leica. It is with great pleasure today that I bring to you Aivia 15 that is focused on faster and easier insight generation powered by AI. I'll first summarize the key new features in this release, then show you in a demo the new features that are available to Aivia subscribers for version upgrade. Aivia has a number of innovations over the years, including the pixel and object classifiers launched in 2017, pre-trained deep learning models for EM, image restoration, and neuron segmentation. Aivia 10 brought us Aivia that learns deep learning-based parameter estimation tool, and in Aivia 11, we brought to you modified cell pose for 2D and 3D detection of cells. In the last 2 years, we brought a number of innovations in rapid succession, starting with Aivia 12, focused on AI-powered soma detection in dense neuronal images, utilizing modified cell pose with a complete workflow for classification based on morphological characteristics and data exploration, including 3D shoal analysis for dendritic density analysis. Aivia 13 marks our focused innovation for spatial biology tools, including multiplexed cell detection in 2D, as you see here, and 3D available in Aivia 14 with automated phenotyping based on intensity and morphological characteristics, and the number of interactive exploration tools, including microcluster dendrogram that you see here that shows similarities between phenotypes, as you see down here, or measurements, as you see up here, as shown by these tree branches, which enable users to explore cell types of interest in their spatial context. With Aivia 15, we lower the technical barrier to entry to AI further by introducing a number of new usability features that will make it even easier for scientists to achieve accurate segmentation powered by AI. Prior to Aivia 15, the following workflow was already possible. Users could view up to 25 channels in 3D, as you see in this spectroplex image, and up to 102 channels, as you see here in the cell dive image that we've tested simultaneously, this is the maximum number we've tested simultaneously in 2D. Then using a number of different AI-based models, accurately segment cells, including multiplexed input, as you see here in this spectroplex image. Additionally, there are specialized detection models for additional structures such as neurons, as you see here, or tracking cells. From segmentation, you can gain further insights about your image by supervised or unsupervised classification, then use a number of data exploration tools, as you see here, as in an example of a dendrogram that shows similarity between different phenotypes and biomarkers for cell types of interest, such as a particular type of cancer cells. Other exploration tools include our violin plot that can plot different measurements or confidence values, as you see here, for different classification methods, along with Welch's t-test results here, up here shown, dimensionality reduction, or various types of other visualization tools such as density plots, as shown here. As part of our ongoing push for continuous improvement, we conduct user insight studies, and prior to our current release of 15, we got a number of different feedback from our users. Positive feedback included feedback such as we have very powerful AI capabilities using the pixel classifier and recipes. We have great, fantastic support, and that Aivia is certainly easier than coding to achieve accurate segmentation results. But the not so great feedback included, and we pay a lot of attention to this, is that oftentimes users needed help to get started. And for novice users, it was difficult to get to the desired endpoints on their own. And to make AI even more approachable than what we already had, we decided to focus on usability features, including having an analysis launchpad that asks users for quantitative endpoints up front to help guide them. Also, this new segment by example tool that asks users to simply paint on cells of interest to deploy deep learning models that are pre-trained. We present users with guided sequence to show users what to do next. And we improved overall speed for our entire 3D pipeline to help you get to insights faster. With Aivia 15, we achieved the following key targets, which give us confidence that we've delivered on our goal of further improving usability. Our users report their need to ask for help to get started was reduced by 67%. Overall processing speed was improved for the entire 3D segmentation pipeline by 69%. And users reported usability improvement in our surveys. Aivia 15 has the following key features. The analysis launchpad that asks users the analysis endpoint upfront, then guiding the users through the guided sequence that is optimized for the endpoint that the user has chosen and the dimension of the images. Segment by example parameter estimator for our deep learning algorithm that is modified from cell pose for 2D, 3D cell segmentation. Overall 3D pipeline speed up to 69%. Updated spatial relation measurements to include batching capabilities. We now also load Leica Live files, including library files, faster by up to 99%. We now enable users to automatically generate reports. And we can now import a number of whole slide scanning files, as well as QPTIFF files. And lastly, we enable users now to export phenotypes to LMD laser microdissection microscopes for multi-wells and other collection vessels with automatic sample allocation. Here are the features that we brought to you to get you from eye to insights easier. We're very excited about this simple painting tool we call segment by example. Users simply have to paint on a small number of cells for Aivia to estimate the parameters needed to feed into our detection model that's deep learning based, modified from cell pose to handle varied morphologies you observe in the sample. Here's an example file from a 3D intestine organoid, where here's an example of what the painting looks like, and I'll show you in a live demo. Here is the preview image from a single 2DZ section, which looks really great here, just based on a small number of cells that are painted. And then all you have to do is run to get the entire 3D meshes generated by simply painting on a single Z plane. And this is the detected meshes that you see at the end. On the right here, we have a bright field image, and the same tool, exact same tool, can be deployed to detect these goblet cells as you see here. So it's quite versatile. Here's an example from this beautiful 48 channel cell dive image with the input channel selected here, Pansy K. Then you simply have to paint. In this example, the red cells show where I've painted different cells of different sizes, as you can see here. And then you can preview, then deploy, and this is the preview result. And the preview window can be resized and deploy to the entire image to get accurate segmentation, as you see here. Once the cells are detected, you can explore the data by using a number of different options for visualization, including different levels of transparency, as you see here, 75% on the left, 0% on the right, or color the object by measurements such as area here or circularity, using different heat map options, as you see here, with different colored heat maps that are available and different measurements you could choose from. If you've ever seen Aivia in the past, you would have seen a powerful tool we launched in 2017 called the Pixel Classifier. The interface looks similar to the deep learning-based segment by example tool I just described. However, the Pixel Classifier has different strengths, as you see here. For example, to isolate a feature of interest, as you see in this Golgi-stained neuronal images. If you've ever worked with Golgi-stained neuronal tissue sections, it's as clear as mud in a 3D view as you see here. What we can do in Aivia is, using the Pixel Classifier, you can accentuate the dendritic features using a line feature filter to isolate dendritic structures, then feed that channel through our automated 3D neuron analysis workflow to get traced neurons, as you see here. In addition, you can perform noise removal. So when you have a very noisy image, as you see up here, using the Pixel Classifier, you can create a new channel to remove noise. Lastly, you can use the Pixel Classifier for multi-channel input masking. For example, when you have a human spleen section that's a bright field 2D section image, as you see here, you can isolate cells of interest, in this case all the brown cells, using the Pixel Classifier with multiple channel inputs to create a new mask channel, from which you can apply the segment by example tool to get accurate cell detection just in the masked channel. Because our segment by example has an implicit understanding of the concept of cells, you get far better result using this workflow than, for example, Pixel Classifier alone. In combination, the Pixel Classifier and segment example together can handle a wide variety of images for segmentation. So please invite us to explore your specific data together with you to see if these or many other types of tools in Aivia would be right for you. Another key feature we focused on this release is the Launchpad that presents guided sequences based on the endpoint. Here's an example from a use case where a user wants to do tracking analysis. Based on that endpoint and the dimension of the images, the steps are shown in this guided sequence, where if a user chooses a detection step, we recommend 3D object tracking as a recipe to use for this image based on the user's endpoint and the dimension of the images to make this entire workflow easier for a user. To provide further guidance, our Launchpad now includes detailed tutorial guides that will help users to delve further into the technical details of a particular method we suggest. For example, for this cell application, if a user wants to study cells including vesicles, then we launch our wiki that specifically explains the workflow for this type of cell analysis to walk you through the workflow, as well as to explain all the details the user may want to know. Aivia is a highly flexible tool, such that until Aivia 14, we presented every tool for users all at once, as you see here. With Aivia 15, we only present to you the tools appropriate for the dimension of the image to help narrow down the choices from many available options we have in Aivia. For more advanced users, it is now possible to create your own workflows that can be customized by inserting a step, renaming a step, insert any function, to save the entire sequence to be shared or reloaded later. As a postdoc, I personally spent many hours compiling reports of the result of my image analysis to share with my supervisor and colleagues. We make this report generation automated in Aivia, where users can create a report such as this one you see with just a few clicks. By default, all the information shown here, such as the image information, metadata, channels list with the appropriate lookup table and pseudo-colors, the object sets summary including counts and percentages, as well as how the objects were created with what type of parameters, are all included. In addition, users can now click on any chart to include any number of charts to attach to the report, which can be exported in Markdown, HTML, and Word doc files for easy sharing of insights. In addition to the number of charts already available in Aivia, we now include a measurement selector functionality that's shown here as an arrow that can help users to flip through measurements for rapid exploration of data. For example, you can have a scatter plot with two different measurements that you could rapidly flip through to change the measurements for easy insight generation and data exploration. You can also select any measurements as a heat map for a scatter plot, as you see here, or create a two-sided violin plot to see distribution patterns of your data for different measurements, as you see here, for different phenotypes. To help you get to insights faster, we have a number of features we've implemented. In addition to the faster LASX loading that I mentioned, we improved the entire 3D pipeline such that you can improve the speed of your 3D analysis by up to 69% based on the total processing time. What that means is that when you have images like this one, that's a 15-plex 3D spectroplex image, you can get to your insights that much faster by using multiplex cell detection in this case, and phenotyper, that's a classification method. We also have created a number of new features that enable more discoveries, with an example here from the new LMD phenotype export functionality. First, we updated our spatial relations such that you can now get measurements such as overlap analysis for different types of analysis, such as the nearest centroid, as you see here, or full overlap as an example, and the nearest neighbor analysis. In this example that I'm showing, you have outlines from the detected multiplex cells that show a heat map from the cyan cells that you see here, with the red cells being the closest and the blue cells being the furthest. This spatial relation analysis, or phenotyping, here's an example of phenotyping using neuronal morphologies, the dendritic arborization as a measure, for example. You can batch, for example, spatial relations and phenotypes, or even channel creation to apply the insight to a large number of images. For example, for workflows such as batching channel creation via image processing, then segmenting cells using a newly created channel, applying a trained classifier, and performing spatial relations can all be batched in a single batch workflow using this button that's now available to you, with a visual that you see here that shows the input and output options for each step. Spatial proteomics is where highly multiplex images can be analyzed for their protein composition and spatial organization, and it was chosen as the journal Nature's Method of the Year in 2024, as you likely have heard. Leica's laser microdissection LMD microscopes are perfectly suited for precise dissection of cells for downstream analysis, and in Aivia 15, it is now possible for detected cells and classified phenotypes to be exported to Leica's laser microdissection microscopes by using auto-allocation of cells to wells or collection tubes using different allocation methods, such as evenly distributing, as you see here, or one set per row, as an example. We have also enabled importing data from virtual slide scanners from Perio, Hamamatsu, Mirax, Philips, as well as Akoya's QPTIFF, so that you can use Aivia for cell segmentation, phenotyping, and downstream spatial analysis that can all be batched. We also bring you additional visualization tools, including binned or bubble scatter plots that can be normalized, as you see here. So here are examples of binned scatter plots and bubble scatter plots that are normalized or not, to show different distribution of cells for different measurements. Lastly, in our continued support of active collaboration with the scientific open source community, we have expanded Aivia's drag and drop functionality for bioimage IO zoo models. So previously, we had launched 4 Aivia DL models converted from bioimage IO models to drag and drop the deep learning models that are converted to Aivia DL directly into the recipe console, as you see in this video. In this example, we have a bacterial model by Establitz Gomez. You simply drag and drop into Aivia's recipe console, and it's applying the deep learning model available on our website that's been converted already to Aivia DL model. And now you have detected results here. And with Aivia 15, we've increased the supported models to 26, which are now available on our website for you to simply download and apply in Aivia. Here is a list of the 26 total bioimage IO models that are converted to Aivia DL models placed on our website for you to simply download to apply in the recipe console. Now I'll move on to Aivia to demonstrate some of the new features. When you start Aivia as of Aivia 15, you'll be greeted with this new analysis launch interface. As described earlier, we propose the optimized workflow for a user based on the user's quantitative endpoint. In this example, I'm going to choose tracking as an example, as well as the image dimensions. I'm going to choose a demo image of nuclei tracking, which is a time-lapse image. As I described earlier, in this time-lapse image, which is in 2D, when I move to the detection step, it already recommends the optimized recipe, which is in this case nuclei tracking, and it also suggests steps for classification, relation analysis, as well as exploration, with the layout shifting to make it optimized for a user. At any point, if a user would like to get back to the launchpad, there's this nice rocket icon that gets you back. In addition, you can get tutorial on how to perform your endpoint by clicking on any of the tutorial links, which goes into great detail about what each parameter means and how to optimally use them. At any point, a user can choose to close this and permanently turn this off if you no longer see a need for this suggestion. Now I'm going to show you an example of segment by example applied to this two-dimensional multiplex image. As you see here, we have a multiplexed two-dimensional image where one channel PanCK-AE1 clearly delineates the cellular membrane. This image has a number of variations that you see for morphologies and size, and I'm going to show you how to apply the segment by example tool that's available under the detection step to accurately detect cells. So I'm going to choose the input channel PanCK. Now I'm going to choose a contrasting color so you could see my painting fairly well. The purpose here is for me to include cells of different sizes to indicate to Aivia what I'm looking for. So I'm going to make the brush a little larger here to paint some examples. First, I'm going to paint some very large examples here and some smaller examples maybe right here and somewhere in between. And the minimum requirement we've set in our current algorithm is 5, just so that we can get the largest, the smallest, and the cells sort of in between. So here are some examples that I'm painting, and I'm not being super precise, and the only thing you have to remember is you don't want to be painting too close to each other, because then the cells will be merged and Aivia will think it's of one large cell. Once I painted 5 here, and then, as you notice, of different sizes, I have a preview window activated here so that I can see what the detection looks like. Unless I define a preview window previously, then it will place the preview window somewhere in the middle of the field of view. And to me, this looks actually pretty good on the first try. So I'm going to apply it to the entire image. So I'm going to run segmentation here for the entire image, and this is the output, and I think it looks actually pretty good here. I'm actually quite happy with it, and there's no training involved here. It's simply parameter estimation, and I'm actually selecting the objects that I've just detected here and adjusting the transparency so you could see the output, and to me it looks actually really good here. And that's a transparency option, and you can also color based on measurement. So in this case, you can color by measurement using area, for example, circularity, and you have many different measurements you can add. So when you go to the spreadsheet, you can have a list of different measurements. So based on morphology, intensity, position, and count, we have a very long list of measurements you could apply. Color by different measurements. You can have fun. You can batch apply this to any number of images, so on and so forth. So this is a super useful tool that we really love. Even those who designed the previous recipes really find this interface super intuitive. Now I'll move on to bright field image It's a fundamentally different type of image as it shows RGB bright field images, but you'll see the versatility of this algorithm. So I'm going to start a new segment by example segmenter where I'm going to use the blue channel as the input, and I'm going to change the color to something more contrasty. I'm going to paint on 5 cells, just roughly selected here and here. 3, that's 3. And this is 4. And maybe this is 5. Okay, then I'm going to do a preview. So the power of this segment by example tool is we have you simply paint so that you can estimate the parameters to feed into the deep learning mechanism. So this is the detected cell. I think in terms of the goblet cells, it looks really fantastic, looks really good. If you would like to remove these brown cells, you can also deploy pixel classifier, which can help to mask based on different channel inputs. I have generated such a channel here that I can also apply. And you can see that using a different channel, for example, using the pixel classifier as a pre-processing step, will remove all those smaller cells and detect just the goblet cells. So you can, of course, in this detected set of cells, you can adjust the transparency, as I showed you earlier, appreciate the segmentation results. They look really good. Again, color by measurement based on, for example, circularity or any number of measurements that you choose to apply here. Now I'm going to move to a 3D intestine organoid image where we see cells delineated by this membrane stain that we can use to apply SBE to. I'm going to choose about here to start my SBE painting. So I'm going to use the input channel of green, get a contrasting color again so that you can easily see where I'm painting. I'm going to make the brush slightly smaller so I can get the smaller cells. So here, for example, here, I'm going to get this one. 1, 2, I'm going to get the smallest one I can find, maybe somewhere around, maybe here. And fairly large ones like this, and somewhere in the middle. Like this one. So notice I'm only painting on a single plane. Let's do a preview here. Let's see how that looks. So when you have examples like this, where you have a pretty good signal here for membrane, and of course, depending on what plane you're on, it may be slightly out of focus. So this is a pretty good representation of what you can expect out of this segment by example. I am happy with it. This looks really good. Notice I painted 5 cells on a single plane. I'm going to run the segmentation to see how well the 3D mesh creation worked in this example. So in this case, we are, so this is the result. It looks fairly good to me. I'm going to go into a 3D view. And there it is. 3D detected meshes that simply by painting 5 cells, we can apply our deep learning model. And I think it works really well for what we would like to achieve, which is to detect cells. Once you detect cells, then there are a number of things one could do. So now I want to move on to a more complex example to show you what type of interpretation is possible. So in this example, I'm going to use this multiplex image with 30 channel input obtained from CellDive. What I've done here is, when we go to the detection step, and this is one of the innovations we brought where we can apply a multiplex input for this rather complex image with a number of different channels for biomarkers that are interactive. So in this example, I have already detected 19,248 cells using a multiplex cell detection recipe where I've specified 8 different channels as input that are delineating the membrane or cytoplasmic area. And when I apply for this image, it takes a little under 2 minutes. In the interest of time, I've already run the analysis and generated measurements. So from here onwards, I want to show you how you can train a classifier or a classification step so that you can actually get insights about your images. First of all, when we zoom in, you could see this is how the multiplex cell detection looks. And I am going to, and you of course can add nuclear information as well. And I am going to apply a classification step here. I'm going to use it as a tumor classifier. And there are a number of different classifier steps that we have built in. The first two are supervised, and the last one is unsupervised automated method of clustering based on phenograph latent. I'm going to use supervised method, and of course if you want to see the difference, there's a difference here that goes into details to help you choose. I'm going to apply the phenotyper to cell membrane and create with 3 classes. So the first one will be a type of tumor called hypoxic tumor. The second will be called normoxic tumor, and the third being normal. And I'm going to use different input here. For this one, the input will be GLUT1. This one will be A1 and GLUT1. And the last one will be PKM2. Now, you, as the subject matter expert of your sample, you'll know which channels to use to distinguish. I've chosen intensity values here, but you can of course go into custom and use morphological measurements to classify your cells. What I'm looking for specifically here is I'm looking for cells that are high in GLUT1 channel intensity to be a part of this classifier. On the other hand, for the second class, I'm looking for cells that are low in GLUT1 but high in A1. Notice I don't have to specify high or low, I just simply need to choose channel or measurement. Once that's done, there are different ways of trainings. For example, one option is for you to visually inspect. So zoom in, I'm going to turn off the nuclear membrane and see which of these cells are, let's say, looking over here where I expect, based on visual inspection, normal cells. You can actually look for cells, right click, and then assign the cell individually to normal tissue. You can select multiple, for example. Or I'm going to show you some cool ways of classifying cells at this point. I'm going to move on to dimensionality reduction, which is a way to reduce a high number of dimensions into simply 2 to help you understand your data better. So in this example, I've selected all the channels except for DAPI, leaving the setting as the default UMAP, and I'm going to generate a UMAP chart. I'm going to make it smaller here a little bit and reduce the charts, and I'm going to resize the image so you could see what I'm doing here. So in dimensionality reduction, you're looking for a nicely separated cluster like this in this embedding that you're seeing. What I'm going to do is I'm going to actually select this particular cluster, and my visual inspection shows me this is indeed, this is not the channel I'm inspecting right now. PKM2 is the correct one, so I'm looking at PKM2 and A1, and I'm going to turn off all the immune cells so that we're not confused, and GLUT1. So there are these 3 channels I'm looking at. So in this example, a lot of these cells that are low for PKM2, which is the red channel like these, are indeed what I'm looking for to classify into normal cells. So a lot of these cells that I can in one go train all these cells to belong to a normal cell group. Another option that I have is, first of all, I'm going to get out of this option. So I'm going to select to get out. Then another option is to have a scatter plot, where this is the cool way to actually flip through different measurements that I was showing to you, so you can explore your data very actively, rapidly, to get insights. In this example though, I'm going to do AE1 and GLUT1, which are the two channels I'm interested in. For the hypoxic tumor, I'm looking for cells that are high in GLUT1 but low in A1, so they would be around here. What I can do is I can basically gate using this rectangular gating method and select those cells, and let's see where they are. They're around here. I'm going to actually select a bit more to include more cells here. So what we're looking for will be cells that are high in this kind of green yellow channel and low for blue. So zooming in closer, I would say some of these cells like here indeed conform to what I'm looking for, so I'm going to assign these cells to a hypoxic tumor. For the second class that I have, normoxic tumor, I'm looking for cells that are high in A1 but low in GLUT1, so I'm going to gate again over here like this. And what I'm looking for is cells that are high in blue but low in this yellow-green channel, and indeed that's what I see here. And I'm going to assign, let's say I spend longer time confirming that these are good cells that I'm going to use for my classes. Then for the third category, I'm going to look for cells that are low for both GLUT1 and PKM2, so that would be somewhere down here. So I'm going to gate based on something like this, and indeed these are the cells that I'm looking for, so I'm going to right click and assign these to normal cells. Now when I get back to my classification, in that short step I've already trained over 7,000 cells that are normal. Normoxic is about 800 cells that I've included, and hypoxic is about 267. So what I'll do is I teach, and you'll see this confidence meter that Aivia has that tells you whether Aivia is confident it can classify. In this case, yes indeed. I'm on a remote desktop, therefore you don't see the cell outlines, but if you were working on your own desktop, you'll be seeing actual cell outlines here. So now I'm going to apply to the cell membrane objects I have here, and I have now created in just a few steps these classes: hypoxic, normal, normoxic, and this very interesting category of cells that are unclassified, which don't neatly fit into the definition I've given Aivia. And that's where discovery can happen. So let's inspect what we have found here. I'm going to turn off the channel here, and I'm going to spotlight all the classes. And what we see is a distribution of hypoxic type of tumor cells here in this pancreatic tumor section. And green is reflective of the normoxic tumor that you see here. Normal tissue, as I described earlier, down here, and all the brown that are unclassified. And this is where interesting discoveries can happen. We can take this section, for example, and apply an automatic classifier to make new discoveries, for example, or we can train a new classifier set with different intensity or morphological measurements. This is indeed a playground for you to explore data using whatever characteristics you are either looking for, in the case of supervised classification as I've shown you, or for true discovery, you can have your data guide you and explore. So here I'm going to now save this classifier to a file so I can show you how to batch this as well. I'm going to override what I previously had. Then I'm going to show you how to do spatial relation measurements. So I'm going to do tumor distance relation analysis where I'm going to look for nearest neighbor from a type of tumor, hypoxic tumor, to find the nearest within 200 micron radius normal cells. And I'm going to leave the default measurement, mean distance, and nearest distance. And I'm going to do this measurement. So when I run this analysis, which is pretty fast, you will see that this distance, and for 19,248, we're doing vertex to vertex distance measurement, and it goes rather quickly. So here we are going to see the nearest distance measurement is already calculated for all the membranes that are in the particular hypoxic tumor category, which is 2,643. Here we can have some interesting visualization options. I'm going to clear the spotlight function, and instead I'm going to go to, in the membrane, we're going to color by measurement here based on the distance, nearest distance, known as tumor distance, where I would like to have the visualization reversed so that the closest cells will be red, furthest will be blue from the normal tissue. So I'm going to display the normal tissue using its own subset color. So here, anything that's closest to the normal tissue here will be red, and furthest will be in navy blue. So this is how you do distance analysis in 2D and 3D between any phenotypes, any object sets, any object types. You can batch this as well. In fact, let me show you how to do that. So I'm going to save the spatial rule. Before we go on, I'm going to show you one very interesting visualization options here for dendrogram, which is when you take the entirety of the phenotyped object set, you can select all the channels that are relevant, and you can generate this dendrogram, which is a really fantastic way to explore your data in depth. Much the same way your family tree will reveal the similarity, genetic similarity between you and your siblings versus distant cousins, this dendrogram shows you the similarity between different measurements as shown in these tree branches, as well as different phenotypes. So for example, you could see the normal cells versus the hypoxic tumor cells have very different intensity distribution for different biological markers. So when we select, for example, the hypoxic tumor, specifically here, you can interactively highlight particular clusters, visualize them, turn on some channels of interest, and inspect the cells of interest in their own spatial context. In addition, let's say you're interested in a particular type of measurement. You can select a measurement, you can sort your clusters based on that particular measurement, you can reset, and really have fun exploring your data on a really deep level to understand what you're observing in your particular data. Now that we've done this, I'm going to show you how you batch. So in this example, you can batch different object detection as well as different spatial relations. If we select this option, every step involved, including detection, classification, and spatial relations, will all be batched at the same time. And here's a visualization of all the input channels and output for our batching process. So you could see there's channel inputs, recipe apply, and also different types of classifiers with all the details that you could see here. And you can also, when you select the relations, basically every step leading up to the relation analysis is batched automatically. So in this example, I'm going to set up a batch process. And what you need to do is you specify which files need to be batched. So for example, in this particular workflow, if I selected these 5 images, it will just in sequence run the batch analysis, export measurements, as well as summary tables, as well as CSV, and all the channels if I choose, and I can run this, which takes a few minutes. And this is essentially how you do this batch analysis from everything, including detection, classification, and spatial relation analysis in Aivia. So if I were to press go, it will just run on its own for under 10 minutes and give you the output you're looking for. So that concludes my demo of all the tools in Aivia. I hope you can utilize a lot of the AI-based tools we have to make more discoveries more quickly. Thank you very much for your interest and watching this demo. Thank you.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, can segment by example handle detecting cells of very different sizes in one image?
It's a great question. So we have built in for this first iteration the ability to handle different sizes automatically without an option for the user to adjust the size range. In the next iteration, we'll allow users to adjust the size range, but for now, we can handle a small amount of size variation in an image simply by painting. The minimum requirement currently is 5. And we recommend users to paint the smallest cells, the largest cells, and the average size cells to give us a range that we can work with. Great question.
Wonderful, thank you. All right, moving along here. Our next question asks, my images have a lot of intensity variations. Would I need to train SBE on multiple images?
Great question. So despite the fact that this looks a lot like a lot of the painting tools for training, such as the pixel classifier, which Aivia has, there's absolutely no training involved. So we're simply deploying a pre-trained deep learning model based on the parameter estimated from users painting, which gives us the estimation for the cell sizes. Currently, we can handle various intensity variations in morphological characteristics for each cells, as well as the size, as I previously mentioned. So it's pretty versatile, and users simply have to paint on a single Z-plane to generate 3D meshes, as I've demonstrated. Great question. Thank you for that.
Excellent. All right, moving along here. Our next question asks, how can I detect cells not only based on nuclei, but maybe nuclei and cytoskeleton, 2 features and 2 channels? Your microphone is muted. I'm so sorry.
Yes, I see that. Not only based on nuclei, but maybe nuclei or cytoskeleton. Yes. So currently, we have a number of recipes to make this happen. Specifically for 2D and 3D, we have a number of different recipes enabling users to take in multiple channel inputs, for example, to detect nuclear cytoplasm in our multiplex detection recipes. We encourage you to send us your data and we can work with you to see what is possible. I would need to see the actual data to see whether we can handle them currently or not. Currently, the SBE interface you saw is single channel and single compartment, but in the next release, we'll enable multi-channel input and multi-compartment output as well. So stay tuned. We have many more exciting variations on this tool that we've just released.
Wonderful, thank you. All right, keep the questions coming in. I see them flooding in, so this is wonderful. Our next question for you asks, can you differentiate between cells that cluster together versus ones that don't? So more empty space in the former, there's no other difference in phenotype or markers.
Yes, great question. So we do have spatial relations interface that I demonstrated. So with that, you could do self to self or self to others distance calculations that are vertex to vertex based. Meaning when you have complex shaped objects, we try to go for the most accurate level of detection such that each cell can have a distance information to another cell type or to its own self. And that can serve as the basis for dissociating the difference between cells that are close together versus cells that are far apart. And you can also visualize that in a distance-based heat map. That can be something of interest for you as well. Again, I'll be happy to take a look at this, and we have a whole team in your region who can help you with assessment. And please, you can go to Aivia website and then ask for a free trial license so that you can try this out for yourself.
Perfect. Thank you. Our next question here asks, how can we detect tumor cells invading vessels?
Yeah, so in this case, what we have is when you have detected the vessel structure, we can do spatial distance analysis and see estimate the distance from the cells, tumor cells to the vessels, and do various types of analysis. You could filter, you could gate. There are several different options that are available in terms of distance analysis. We can also do things like nearest 5 cells or nearest distance. We have many different possibilities. We encourage you to, again, contact us and have us evaluate your data. And I think there's another interesting question there.
Yeah, this next question that we have is asking, is the cell painting tool able to detect a variety of cell morphologies, such as elongated cells in addition to circular cells in the same tissue section?
Great question. So as I mentioned earlier, we have modified a very powerful generalist algorithm, the cell pose algorithm, and we've modified this so that we can adapt it to different situations in 2D and 3D. And because we are based on cell pose, any morphological characteristics you see in cells that are similar to the ones used in the training for cell pose. And if you recall, in the actual model, the authors trained the cell pose on many different variety of cell types, including some elongated ones as well as some cells with irregular borders. They even trained using seashells and different types of objects that are not cellular at all. So we do detect some elongated cells. We are currently experimenting with training additional models that are able to handle even greater variety in terms of morphologies of the cells. So some elongated cells, yes, I would say so. We can handle many different types of cells that are not only circular and round, but somewhat elongated as well. Great question.
All right, thank you. And it looks like that is all of the questions that we have today. So I do want to thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.